Okay, so now we move over to another delegate, Ms. Claude Hamany Djande, who will speak on a metabolomics approach and chemometric tools for differentiation of barley cultivars and biomarker discovery. Please, you are welcome to share your presentation then. Marie, you can stop sharing. And I'll hand over to Hamany Djande from the University of Johannesburg. Just a reminder to our presenters to please just show your face or introduce yourself. And once you start your presentation, you are then free to sort of mute your video. Yasmin, hi, we're able to see the presentation, but I don't hear you. Okay,
I think it's fine now.
Yeah, I hear you now. Please go ahead.
Can you see the presentation?
Yes, I can.
Okay. Okay. Sorry. This one.
Just put it in, I think, presenter mode. It's showing your slides and the notes as well. Yeah.
All right. Is that fine now?
Yes, thank you so much. I don't know if you just want to wave and show your face, perhaps.
My face.
Just unmute the video for a second or two.
Okay.
Okay. Thank you.
I'm Claude. All right. Okay, I'm going to present to you, my name is Claude. I'm going to present to you a metabolomic approach and chemometric tools for differentiation of barley cultivars and biomarker discovery.
Sorry, Yasmin.
Yes.
I'm not sure if you have two devices on, perhaps. There's a little bit of sound that's echoing.
Okay, I've noticed this. Is that fine now? Is it fine now?
Yes, it's much better. Thank you.
Okay. Okay, I'm going to present this part of my PhD work. Okay, sorry. Oops. Okay, so crop production can suffer from diverse environmental pressure, being abiotic or biotic. These pressures are huge setbacks in the goal of meeting the increasing demand for crops in general and barley grain in particular. Aiming then to increase crop productions, different strategies are often explored. An example is the development of new cultivars presenting traits or phenotype of interest through plant breeding techniques. Okay. These cultivars are developed for improved environmental stress resistance these cultivars are developed for improved environmental stress resistance, yield, and quality traits. Although tremendous advancements have been made in breeding cultivars, one important challenge remains to develop a cultivar with all the desirable phenotype. Such a task is difficult because of the pleiotropic effect of some genes, the structures of the genetic population, and the linkage that exists between genes on the chromosome. In addition, the change of a selected trait may result in the anticipated or unanticipated change connected with the, connected with the trait of interest in the desired or the opposite direction. Okay. Although progress in understanding the system biology of agronomic traits has been mainly attributed to genetic and genomics, recent plant breeding approaches take into consideration important information provided by the multidimensional data related to epigenome, genome, transcriptome, proteome, and metabolome that collectively impact the phenotype. In fact, there is a close relationship between the genotype, the phenotype, and the environment. Phenotype are influenced by the genotype, the environment, and interaction between the two. Inversely, the interaction between the phenotype and the environment can also affect the genotype. Moreover, the variation of the genome affects the phenotype through a series of downstream events on the transcriptome, proteome, and eventually the metabolome. The influence of the genotype on the phenotype facilitates the use of DNA-based markers in plant breeding practices for the selection of important traits. The challenge here is that some genes may be present in the biological system, but not fully expressed, and therefore result in ambiguous selection of the desired traits. This can be overcome by the complementary use of metabolite-based markers, given that plant metabolome constitute a bridge linking the genotype and the phenotype. So in this study, an untargeted metabolomic approach based on ultra-high performance liquid chromatography hyphenated to high-resolution mass spectrometry was employed for chemoprofiling and discrimination between 5 genetically related and unrelated cultivars of barley from the Western Cape rainfall region of South Africa. Okay, the study was designed as such. 5 cultivars of barley, Hessekwa, Agulhas, S16, Elim, and Hessekwa, were cultivated in 3 biological replicates under a controlled environment. After 21 days, samples were harvested and extracted with 80% methanol. The extracts were analyzed on LC-MS system in both positive and negative ionization modes. Using a binary solvent system, the data obtained from the analytical platform were pre-processed and pre-treated using the MassLink software. And for chemometrics and statistical analysis, Steenkamp and Metabolomics softwares were used. This workflow allowed to provide a differential metabolic profile of the cultivars under study. Sorry. Okay, the base peak intensity chromatograms of the leaf and root samples are represented in these figures. The leaves on top and the roots at the bottom. The visual comparison of chromatographic profile revealed more distinguishable quantitative as compared to qualitative differences between cultivars. Examples of such variation are highlighted with dotted rectangles. We can also note that a wide range of metabolite ranging from polar to non-polar way could be depicted on the chromatograms. Okay, aiming then to holistically understand the cultivar-related differences, data processing and chemometric analysis of the complex and multi-dimensional data were required. 2, unsupervised chemometric methods, principal component analysis, as well as hierarchical clustering analysis were performed and provided an overview of patterns and trends therein. The PCA scores plots showed clear sample grouping with distinct differences between extract from leaves and roots, indicating cultivar-specific variations, as well as tissue-specific differences. In addition to PCA, HCA displayed the same pattern observed in the corresponding PCA, and more defined subclustering was underlined. Okay, before the investigation of the differences among the cultivars, a global profiling of metabolite was performed to provide a general background on the chemical composition of barley leaf and root extract, irrespective of the cultivars. So the annotation was mass spectral based, as it can be seen in this example. So in this specific case, the fragmentation pattern of individual metabolite could not be clearly obtained due to the co-elution observed here. However, these compounds were annotated as rhodantine A, B, C, and D, based on the mass difference of 30 Dalton between the different rhodantines, as well as the fragment ion of 131, 114, and 157 corresponding to agmatine derivatives. These compounds, I chose these as an example because they are barley-specific phenylamide, and they have important antifungal properties. We know, I didn't mention, but barley is mostly used in South Africa for its malting purposes. So these compounds also possess that astringent aftertaste effect that we can test on beer. Rhodantine A recently was found as a potential inhibitory of COVID-19 main protease and RNA polymerase. So, of course, these rhodantines were not the only metabolite annotated in the study, except for adenine identified with the help of authentic standard. 74 metabolites were positively annotated. Among these, 69 were found in the leaves and 30 in the roots, together sharing 24 metabolites. Phenolic acid and derivative were the major class of compounds in the extracts from both leaves and roots of barley cultivars. In leaf extract, this was followed by flavonoid and fatty acid. In the root, this was phenolic acid were followed by fatty acid. Only one flavonoid, saponarin, was annotated in the study. Organic acid, amino acid, and alkaloid present in both tissue type represented altogether the smallest percentage of metabolite. In addition to differences observed between tissues, plant tissues, it was possible to also depict differences across of these classes of metabolite across cultivars. In leaf extract, for instance, phenolic acid, organic acid, amino acid, and alkaloid were more prominent in Elim as compared to Agulhas. Okay, The diversity observed in the class of annotated metabolite was remarkable when investigating individual metabolite across cultivars. This was infographically illustrated in heat map format, highlighting the differential relative concentration of specific metabolite within each metabolite class. Okay. So to select discriminating metabolite between the cultivars, a statistical tool such as PLSDA was performed. These compounds were selected as discriminant metabolite, and only the ones with a VIP score higher than 1 were selected. As an example, in the leaf extract tryptophan ferulic acid exhaust ordatin A and its exocylated conjugate were more prominent in Agulhas as compared to the rest of cultivars. Except for isovitexin 2, isovitexin derivative higher in S16, Erica had the highest level of all discriminant flavonoids. In the root, except for saponarin, more predominant in Erica, Agulhas presented the relatively highest level of discriminant metabolite. Hessekwa, on the other hand, had the smallest relative concentration of metabolite. Okay, in addition to PLSDA, OPLSDA was performed and as a binary classification method and provided with potential signatory metabolite corresponding to each cultivar. 26 metabolite were annotated as discriminant metabolite. And yeah, so in the leaf tissue, for instance, comparing Erica to other cultivars, Agulhas, isovitexin and isovitexin 2O arabinose, isomer 1, were positively correlated to Erica compared to the rest, and the isovitexin 7O rhamnoxidone glucoside was negatively correlated to Erica. Okay, in the leaf tissue, for example, sorry, still on this, sorry. All these metabolites annotated in the study as discriminant metabolite or not are involved in plants' important processes. This figure is the representation of the interlink pathway summary showing the biosynthesis and participation of all annotated metabolite in barley leaf and root tissue. We can see their involvement in the alkaloid phenylpropanoid fatty acid biosynthesis, to only name a few. Okay, so irrespective of the cultivar tissue, of the plant tissue, phenolic acid were the major discriminant metabolite, followed by fatty acid and flavonoid. Organic acid, as well as amino acid and derivative, were more tissue dependent as they were only found discriminant in leaf extracts. Plant metabolite in their diversity can be associated with several agronomical important phenotypic traits. The quantitative and qualitative occurrence of these particular metabolite is a prediction of specific biological state. These metabolic biomarkers can be characteristic of crop performance traits, such as grain or tissue yield, storage properties, morphology, nutritional attributes, sensory, and the list is long. These are important attributes in plant breeding practices. So in conclusion, recent advances have confirmed a relationship existing between genetic variants and metabolite that could be useful for metabolic engineering across various plant cultivars. As essential component of plant metabolism and phenotypic determinant, the investigation of metabolite is important for the understanding of the biochemistry underlying plants' physiological responses. As such, it contributes to the interpretation of interactions and connection which exist between plant system and the environment. The study showed that metabolomic analysis and chemometric tools employed were effective to observe variation between cultivars that are genetically and non-genetically related. That are genetically related and also not non-related. Okay, I'm covering cultivar-related metabolite or a group of economic attributes would provide additional knowledge that can be useful for breeders in the selection process and development of new cultivars. Although metabolomics provide very insightful information, the combination with other biology, system biology approaches constitute a powerful strategy towards a comprehensive understanding of a plant. So this work was published in this journal. These are some of the references that I use to make this presentation. I would love to thank these amazing people. Professor Dubery, my supervisor, Dr. Tugizimana, Prof. Piater, Prof. Steenkamp, my co-supervisors, and these organizations. Thank you so much for listening.
Thank you so much for that insightful talk. I'm going to quickly move over to a few of the questions. I know we started a little bit late. The first question is from Sizi Mhlongo. With the Hortidine A activity against COVID, are beer drinkers expected to have reduced symptoms compared to non-beer drinkers, in your opinion?
I think it's too early to say anything about it.
Sorry?
I think it's too early to say anything about it, but again, it's only one compound of barley that was found successful as inhibitory against those RNAs, against those the COVID. But the fact that it's only, we all know that the beer will have more than that compound. So it's a bit, it will be difficult to say and probably early to make a conclusion toward it.
Okay. The next question is from Abubakar Hassim. The question there is, are these plant metabolites a phenotypic trait, or do they show an interaction between genetics and the environment?
Excuse me, can you come again?
Are these plant metabolites a phenotypic trait, or do you think that they show an interaction between genetics and the environment?
Okay, at this point, I think we can say that they are phenotypic traits. They have been found to, in several studies, to be probably upregulated against when treated with under specific conditions. But because we haven't really studied a specific environment, it's difficult to say that they are the result of any given environment, but we know that those metabolites are metabolites that are important as proper phenotypic traits and can be maybe targeted or increased in terms of production if desired.
Okay. I'm going to post one more question to you just in the interest of time. Were these sample crops grown in the same region or area, or are they from different field areas, or are they greenhouse products? So that's the first part of the question. So were they grown in the same area, different field areas, or are they greenhouse products? The second part of the question relates to the extraction solvent, because it can introduce bias for the phenolic or flavonoid compounds. Was that the intention? And I see there that someone else added onto that question, asking whether the solvent, should the solvent not have been 5% ethanol as in beer? So first of all, just where were they grown, the area where they were grown, and then about the extraction solvent.
Okay, they are for the first question. Plants were grown in a greenhouse. They were under very controlled environment, but they are normally cultivated in the Western Cape region of South Africa. So yes, the solvent definitely impacted the type of metabolite that we extracted. With a different solvent, we'll probably have a different result. And that's the complexity of metabolomics, is that we need to do many extractions, different extractions to have a greater coverage of metabolite. But in this case, we chose 80% methanol because we wanted to have that wide range of metabolite, as we could see also on the chromatograms, ranging from really from polar to non-polar compounds. And we could really get them, we could really get very polar compounds and some of the non-polar compounds. So yeah, that was the purpose of the 80% ethanol. But now the 5% for beer, the intent was not really to stay in the same context as working with beer. Barley has more to it than just the use in the maritime industry. It can also, as we see now, be involved in any different area. So we couldn't just focus on that 5% that is normally used in beer. Okay.
So in the interest of time, there's still a question there, I think by Prof. John Becker, about the use of leaves and roots versus seeds. And also one from Francis Maiba that says, would you predict how these metabolites would express in different environments? If you could maybe, just in the interest of time, please go to our chat room and maybe just address some of the questions that has not yet been answered.